Today I'm going to talk to you about certain biomarkers measuring the activity of genes and how they may have applications to hatcheries. For example, measuring smoltification or seawater preparedness activity in genes in juveniles prior to release. My name is Amy Houde, and I'm a postdoc at the University of British Columbia and with Fisheries and Oceans in the Molecular Genetics Lab. Today I'm going to talk to you about how we identify candidate genes or biomarkers, our validation with new samples including Fraser River sockeye and Chilco salmon smolts, and future directions of research. Also at the end of my presentation, I hope to have a discussion about what may be other potential biomarkers that could be useful to hatcheries. Salmon declines are associated with lower survival in the early marine environments, such as estuary environments that are transitory. I'm proposing a tool that may improve the health and condition of hatchery fish, increasing survival. And for example, in hatcheries, smoltification can be traditionally measured using such measurements as changes in behavior as well as changes in body coloration, such as this increase in silvering in coho salmon. However, seawater preparedness or smoltification can be more precise using the activity of genes. So this is a tool that has an opportunity to optimize culture and release strategies. The tool can also use non-lethal gill samples because very little tissue is required. Identifying candidate biomarkers. So at the DFO Molecular Genetics Lab, we combined as many studies as possible on the activity of genes. We included many species, including sockeye salmon, rainbow trout, Arctic char, and Chinook salmon, amongst others. We included many groups, anadromous and resident groups, pre-smolt, smolt, and post-smolt groups. And without going into the heavy statistics, if you're interested in this, we could talk afterwards. We identified clusters of genes or biomarkers that can separate groups. For example, this figure displays a cluster of genes or biomarkers that can distinguish individuals based on the smolt scale. And this cluster of genes or biomarkers, it's robust across salmoniid species. Our next objective was to produce a universal assay to measure the activity of genes for every gene within the candidate biomarker clusters. And what I mean by universal assay is this would be one assay that would work across all species or as many salmonid species as possible to measure the activity of genes. To meet this objective, we collected genetic template for many species. We have genetic template on sockeye, coho salmon, Chinook salmon, Atlantic salmon, and brown trout, and it's currently housed in a large database. So now we have universal assays for candidate genes related to viral disease development, thermal stress, chronic hypoxia stress, and smoltification with links to seawater preparedness in juveniles or freshwater preparedness when considering returning adults. So now we're at the stage of validating our candidate biomarkers using new samples. Here I'm going to talk about viral disease development. So this is work by Christy Miller. And these are host gene biomarkers that are specific to RNA viruses and host-specific biomarkers that are related to bacteria infections or other classes of parasite infection are in development. So for sockeye salmon, there were 39 candidate biomarkers that can separate a disease state where the pathogen is present and the sockeye is showing a genetic response from a latent carrier where the pathogen is present but there is no genetic response. The 39 candidate biomarkers were validated using new samples from 5 controlled studies and the Fluidime platform. So the Fluidime platform can measure up to 96 biomarkers in combination with 96 samples within the center of the chip. We also examined the candidate biomarkers in terms of a natural context using samples from out-migrating smolts from Chilko Lake. So here are the results for Chilko Lake. This is a figure displaying 213 individuals. And the figure is a multivariate representation of host gene or sockeye salmon genetic response. And the different colors are the different pathogen loads for the virus IHN. So in this figure, there to the left, there is a cluster of fish that are showing a high pathogen load as well as a high strong genetic response, whereas the individuals that are latent carrier or do not have high loads of the virus are clustering more to the right. What's also interesting is that towards the middle, there are some individuals that are showing a genetic viral response, but they do not have high loads of the IHN virus, and they're being examined further. And without going into much statistical detail, we were able to achieve this type of separation using only 9 genes. So with statistical analyses and more information, we were able to reduce the number from 39 candidate genes down to 9 genes in order to achieve this type of separation. And we can also do this type of analyses for other pathogens that may be important to hatcheries where we quantify the amount of pathogen and look at the fish genetic response. Thermal and hypoxia stress. So this is work of my colleague Arash Gozar. And thermal stress issues are likely to be more prevalent in the future with global climate change, increasing water temperature. Chronic hypoxia, or exposure to low dissolved oxygen, has received lesser attention, but it can be an important issue for certain environments such as estuaries if there's increased nutrient loading, lower water discharge, or an increase in algal blooms. There were 39 candidate biomarkers for thermal stress, 14 for chronic hypoxia stress. The thermal biomarkers are being validated using sockeye adults from the Fraser River that were exposed to polar temperatures. And our hypoxia test samples just arrived, so I don't have any results to show for that. These are the results for Fraser sockeye adults. So these are the top 4 genes that are showing relationships of the exposure of temperature. And there's 3 genes that have positive relationships with temperature exposure, and there's 1 gene that has a negative relationship with temperature exposure. The serpin and the heat shock protein genes are important for stabilizing and repairing proteins in the body, whereas elongation factor 2 is a critical factor for protein synthesis in the body. And this is just the beginning of the analyses. With more information, we'll be able to reduce the number of genes, similar to how 39 candidate genes were reduced down to 9 genes for looking at a viral disease response. Now moving on to my work, smoltification. So I identified 46 candidate biomarkers related to smoltification, and right now I'm working with my top 19. Last year I collected validation samples of coho salmon from 2 hatcheries, sham salmon from 3 hatcheries, and sockeye salmon from Inch Creek hatchery and the native Cultus Lake. So these samples were collected monthly over the typical smoltification period from late fall to spring, and have hatchery and wild groups that are represented. So here's a figure for the coho salmon, and these are the top 4 genes that have relationships with monthly development. And they all have positive relationships with the month of development. The first 2 panels, carbonic anhydrase and cystic fibrosis, these are ion exchange genes, and they're likely preparing the gill for going to an ion excretion stage for when the fish enters seawater where more ions are prevalent. The hemoglobin gene, this is important for oxygen acquisition in the body, and typically in marine environments, the oxygen concentration is lower relative to fluvial freshwater environments. And the 60S ribosomal protein, this is a protein synthesis gene that's believed to be related to body growth in salmon. So I should mention that the Chinook salmon pattern is similar for these 4 genes, and since I'm at a sockeye conference, I didn't have the Inch Creek cultus sockeye samples ready in time, but they're on their way. Some of you may be wondering about the sodium-potassium ATPase gene, which has been the focal point of many osmoregulatory studies. So I did examine the precursor for the genes that are related to the saltwater isoform, which is the B isoform at the top, and the freshwater isoform, which is the A version in the bottom panels. So indeed, these genes do change over the course of monthly development, but the change is not as drastic as some other candidate biomarkers that I am considering, such as those that I showed on the previous slide. And what is also interesting is that there's a larger change in the expression of these genes when the fish is actually placed into seawater, as seen with the Chinook salmon. There's an upregulation of the saltwater isoform when the fish are in seawater, and a strong downregulation of the freshwater isoform while the fish are in seawater. So one caveat with the smoltification work is I cannot relate the gene expression values to actual values of seawater preparedness, such as survival or body growth, without doing a controlled study. So here's a tentative plan for a controlled study, and it's combining the work of rash on the thermal and chronic hypoxia stress genes with my osmoregulatory gene work. And in the plan, we would like to examine 3 temperatures, 2 oxygen saturations, and 3 salinities in all possible combinations. And doing this, we would be able to identify the best groups of biomarkers for separating single stressors from multiple stressors. And at the bottom here is just a rough schematic of a possible hypoxia setup. So we'd like to do this over the smoltification period, when fish would likely be more susceptible to thermal and hypoxia stressors. And we'd also like to examine this in adults, considering the thermal and freshwater preparedness context. So in conclusion, we are proposing a tool to improve the health and condition of hatchery fish. And this has opportunities for optimizing culture and release strategies. And this could also be used for fish after release from the hatchery, because this tool could be used to see how salmon are acclimating to the environment, such as the early marine environment where survival has been an issue. Also looking for ideas on what might be other potential genetic biomarkers for hatcheries, and also looking to gauge the international interest in getting this type of information. So in acknowledgement, I'd like to thank my colleagues Christy, Arash, Scott, Tony, the Salmon Enhancement Hatcheries, Vancouver collaborators David, Jamie, students Claire, Olivia, and the Genetics Lab. And also like to thank the funding sources, UBC, Fisheries and Oceans, NSERC, Pacific Salmon Foundation, MindTax, and GNOPC. Thanks everyone.